Okay, good morning, everyone. We're going to start the second day of this colloquium. And it's a great pleasure to have to start this morning with Jonathan. You know, Jonathan was here for several years, and he was too good for us, so he has moved to Brendeis. And we are pleased that you can come back today, Jonathan, and tell us about your modelization studies. And also some biology, right? I mean, real biology. Yeah.
All right. Thank you very much. Thank you very much, Alain, for inviting me. It's a great pleasure for me to come back here. And actually, all the work I will present today has been done here in the Center for Interdisciplinary Research in Biology at the Collège de France. And it's the result of several collaborations, including collaborations with Alain Prochiantz, Alessandra Pirani, and Shenju Xu. And it's mostly the work of two PhD students, Cristobal Quinao and Yi Quick, who is finishing her PhD. So essentially, I wanted to start with a little bit of what I do in research and how the question of neurodevelopment arises in my research. So I'm an applied mathematician. I'm interested in the interplay of structure and function in the brain. So how the structure of the connections has an impact on brain activity and in turn on behavior. So, and I study different types of models that all involve time. So I study dynamical systems to understand the firing of single cells. But I also study the large networks of neurons, and I'm particularly interested in noise and how large neural networks can function in the presence of noise. And also I analyze with different colleagues the structure of functional architecture of the brain, trying to understand how specific organizations, here for instance in the visual cortex, can allow processing of the input. And also related to that, of course, is how does the brain acquire its function through development. And to study that, I discussed a lot with experimentalists, and I will present you two questions related to homeoprotein and development. The first one has already been introduced yesterday and several times. It's the formation of well-defined brain areas in the brain or here in the neural tube. And the model I will present you will investigate the possible role of non-cell autonomous function of homeoproteins in the stabilization of boundaries and the regularity of boundaries between different functional areas. And if time allows, which I hope, I will present you a second model, which acts at a smaller scale, and which is aimed at reproducing the sequence of divisions and differentiations of progenitor cells, yielding to a specific organization of the number of neurons in each layer. And the main question here would be to understand whether if we can infer from a given phenotype, either in the wild-type mice or in mutant mice, if we can infer from this phenotype the precise timing of these divisions and differentiations. So let me start with this first project, which is modeling homeoprotein diffusion and the boundary formation. So this is a joint work with Alain, and this is Cristobal Quinao, and you can see that they've been thinking a lot to this problem. And to introduce it, actually, I decided to cite the paper by James. So essentially, the question, which I will repeat today, but it's been discussed yesterday, is how from graded cues, which are gradients of morphogens, do we end up with a very sharp and precise organization of functional areas with well-defined boundaries and a discrete patterning. And in this review by James in 2006, I think the questions are very well formulated, so I decided to cite it. So it's so crucial to understanding this differentiation, this switch between graded cues and discrete patterning. We need to understand several aspects. So how this information, the positional information, is integrated intracellularly. So I will not discuss about that, but I will discuss questions 2 and 3 that are raised in this paper, which is how a continuous gradient is transformed into discrete changes in gene expression that ultimately determine the choice of cell fate from available alternatives. And very important also for what I will present is how does this signaling accommodate fluctuations in biological conditions to achieve the necessary robustness required for accurate developmental patterning. And so I will try to make more explicit how I understand these fluctuations of biological conditions, and then we will test that in the model. So I would say, we will try 4 aspects of these fluctuations, which are variations of the level of morphogen that is sensed at the level of each cell, variations in the initial patterning, initial concentrations of transcription factors in each cell, the heterogeneities in cell parameters, namely the differences between the way the different cells process the information, and also the phenomena that are associated with the emergence of new cells and the death of new cells. And I will try to understand how the model that we're going to look at accommodates these fluctuations. So of course, when discussing about morphogenesis, I think we cannot avoid a slide on Turing. So I think one of the most important models for patterning is this paper by Turing that showed actually an instability, a pattern formation instability in tissues when you have the interaction of different actors, an activator and an inhibitor, that diffuse across the tissue. And so essentially starting from a homogeneous solution, these systems, these competition systems, form patterns when the diffusions of the activator and the inhibitor are very different. So when the activator has a local diffusion and the inhibitor has a more remote diffusion, you can form regular patterns. So these are examples of regular patterns. And these patterns are interesting because they have smooth boundaries and sharp, as we can see in the nervous system. But one aspect of this is that actually the precise pattern that is formed is unpredictable. I mean, it depends highly on the initial condition, and it may change when you have heterogeneities. So this is the main, I would say, the main drawback of the model for our purposes. And there is another very important model in the compartmentalization of the nervous system, which is Volpert's model, the French flag model, which actually takes into account the fact that this differentiation occurs within a morphogen gradient. And this morphogen gradient provides positional information to each cells, and the different cells respond in a different manner to levels of concentrations of this morphogen, and they are differentiated into different types of cells. So this model has the advantage to provide relatively well-defined boundaries and locations, which are associated with the threshold. But the main issue with this model is that you may have, when you have noise, either in the cells or in the way the morphogen gradient is sensed, you will have noise in the boundary. So the overall position of the boundary is well defined, but it's relatively irregular. So, well, to start this work with Alain, we discussed about what are the main responses of homeoprotein two morphogen gradients. And so what Alain taught me was that homeoprotein are very particular transcription factors that first have an autocatalytic activity. They are actually their own promoter, so they amplify their own expression. Different sets of homeoprotein may repress may repress their expression, either by a direct inhibition or by competing for genetic material or competing for space saturation within a cell. And also, importantly, I would say, for our model, is that these transcription factors also have a non-cell autonomous activity. They have the ability to exit the cell nucleus and exit the cell to be internalized in neighboring cells and be expressed. And this is extremely local. This is a very local effect that acts at a range of 1 cell rank to 3 cell ranks. So it's extremely local. And it is a question whether this very small diffusion can have an effect on the emerging pattern. So essentially, if we want to put these three aspects in equations, so I'm sorry, I will show some equations. I will develop a model that only takes into account these three aspects and investigate its dynamics. I start with a simple model, and then I will go for a more abstract model. So the simplest model we can think of that incorporates these three aspects is the following. So it governs the rate of change of the concentrations of the different cocktails of morphogens, of homeoproteins, let's say A and B. And the rate of change will be proportional to the morphogen gradient, the positional Q. So a gradient of morphogen will stimulate the expression of A, and another gradient will stimulate the expression of B. So this is the first term. So the rate of production of A is proportional to this gradient. We have also an autocatalytic activity. So the more A you have, the more you will create A. So we have this promotion here term, and then we have mutual repression, and in that case, I only considered a saturation effect. When A plus B, so the total concentration of morphogen is too high, we will have a saturation with a coefficient SA and SB. So the system is perfectly symmetric. I only discussed A, but B is exactly the same. So this has the cell autonomous this represents the cell autonomous responses. And what we add to this model is a small diffusion. So one cell will send transcription factors to neighboring cell, and it will receive also from these neighboring cells transcription factors. So this translates into this diffusion operator here, which I see as a discrete operator. It's not a differential in the position. It's just this process that I'm writing delta A. And sigma is a rate at which this process occurs. And as I said, since the diffusion is very local, we may think of sigma A and sigma B as being very small. Okay, so this is basically the model. And let's see how this model reacts. So what it does. So the first thing is that it has two modes of behavior of this simple model. So first, when FA is much larger than FB, so if we are in a region close from the morphogen source, for instance, one of the transcription factors will be highly promoted, and it will strongly repress the other one. And the final state will have a high concentration of A and no B inside. So we have a winner-takes-all behavior near morphogen sources. But when the two, so near the boundary, we will have an ambiguous region. When FA is comparable to FB, well, either A can be expressed and B not expressed in the cell, or the contrary. And this actually depends on the initial condition. if you start from a very high concentration of A, for instance, this A will quickly increase, and it will overwhelm and repress B, and it will lead to the similar state. So this is essentially heuristically what this model does. we can find the solutions to this model because it's so simple, and we indeed find two branches of solutions. One where A is expressed and B is equal to 0, so this red branch, and this equilibrium is stable near here 0, which is the source of the morphogen for A, and it becomes unstable when away from this source, and symmetrically for B. And we have in this region, in this gray region, an ambiguity where both states can be reached and can be stabilized. And here is a plot of these equilibria. So in this region of high FA, we have a stable equilibrium for A and 0 for B, and every trajectory will go to this fixed point. Same thing for in the region of very low FA. So every cell will differentiate into a B cell, and whatever the initial condition. But in the ambiguous region, we have a separatrix that is associated actually with another unstable fixed point. And depending on the initial condition, if you start from the pink region, you will differentiate into A, and that means that you have an asymmetry in the initial concentrations with another representation of A. And in the contrary, you will differentiate into a cell whose fate is B. So essentially, this is problematic because if we start from a low level of concentration of A and B, this separatrix actually reaches 0 here. And so small fluctuations of this concentration will yield to highly different states. And if we start from random initial conditions, or with slightly patterned initial conditions, we end up with boundaries that are not regular, as in the Volkmert model. So here we have a lot of noise at the level of the transition. And actually what happens here depends on the initial condition and maybe on a pre-patterning also. So if I start with an initial condition with a little bit more of A here in the center, this will remain forever in the pattern. But now if we add a little bit of diffusion, so here it's the coefficient sigma A is relatively small. It's about the scale of one cell distance. What happens is that for these cells here that are in the middle, for instance, for one white cell here in the middle of a black region, what will happen is that this cell will receive from diffusion a lot of the transcription factors that are lacking. And this will have a dramatic effect in regularizing the boundary. So even at this very small level of diffusion, what we have is essentially no effect within the ambiguous region, and a well-defined boundary which does not depend on the initial condition. So the final state no more depends on the initial condition. And so we did also this simulation in another topology that looks like the more of the neural tube, and of course we have exactly the same phenomenon because these principles that I showed you do not depend on that. So we have salt and pepper boundaries in the absence of diffusion, but with very small diffusion we regularize the boundaries. So essentially this simple principle ensures a separation into two compartments in response to graded cues, and the diffusion tends to regularize boundary and make boundaries independent of the initial condition. So now what I will look at is, does this diffusion also accommodate for other sources of fluctuations besides the randomness of the initial condition? So we tested, as I said, we tested three aspects of these variations. First, fluctuations in time and space of the level of morphogen that is sensed by the cells, these FA and FB. Now instead of being, so this is position and this is time, and I'm considering that actually this concentration is stochastic, it follows essentially one profile, one decreasing profile, but in time it fluctuates as a Brownian motion, and it's independent location per location. So so different cells, neighboring cells, do not have the same noise, and this noise varies in time. And so with a noise on the order of 20% of the maximal level of the gradient, what we find for the patterning is essentially no effect. So we have a boundary that is relatively stable, and here I plotted the full solution, but if I plot also the locations of the cells, the size of the cell that I consider in this simulation, essentially the boundary, even if it wiggles around, does not really goes beyond, does not move by more than one cell rank. So diffusion stabilizes the boundary even in the presence of noise in the gradient. We also consider the heterogeneous cells that would have different values for the maximal rate of production, the autocatalytic parameter, and the saturation parameter. So what I did was taking for all these parameters a random value centered at the value I considered before, but with a variance. So each cell is a little bit different. And essentially what you will have is that you will have your concentrations. So in the dashed line, we don't see really well, but in a dashed line is for homogeneous fields, and the full line, the solid line, is for this random model. What we have is small fluctuations here, but essentially the boundary is always the same because the solution is quite sharp at the boundary. And we computed for 500 realizations of this process the location of the boundary with 20% of noise in this parameter, or 100% of noise, like a variance equal to the mean. And essentially we move of about five cell ranks by changing by 20% all parameters, and by less than 10 cell ranks in the 10% in the case of 100% of noise. So this system is relatively stable also to this variation. And the last thing we tested is how the system responds to cell division and cell death. So the way we modeled that was to have a number of cells that changes in time, and we have creations of new cells, and the two daughter cells share the content of their mother cell. So that's why you have jumps, because the content of these cells, dividing cells, goes to half of the value. And so we did this simulation, and we made the statistics, and again, the location of the boundary, although the solution may change in the differentiated region, the boundary actually barely moves in response to this process. Once it is set here, it barely moves, even if we have cell division or cell death near the boundary, and that's what we see here. So a conclusion from that is that this simple model of Homeoprotein yields multi-stable system in the absence of diffusion, and even weak diffusion can regularize the boundaries and can stabilize this boundary in face of noise and perturbations. So this was a very simple model. The question is how general is this observation? And so that's a work that we did after that, a more general theory of this kind of competitive systems in the presence of graded cues and small diffusion. And actually what we found was relatively surprising is that actually, although with zero diffusion you have a very irregular boundary, the limit when the diffusion goes to zero of the system with diffusion is very stable. Actually, the less diffusion you have, the sharper the boundary and the more stable it is. So to show that, so I will be very brief on the mathematics, but I just want to state the result. So we looked at a class of systems that have small diffusion, so epsilon is small here, and that have the main properties that I discussed about for homeoprotein, namely the rate of production of A is driven by a gradient. So essentially, this rate of production for fixed values of A and B is decreasing with respect to the position X. And we only assume competition, which is actually the more B you have, the less production of A you have. And symmetrically, we do the same assumptions for B, except that the gradient for B is increasing. And for all these models, actually, what you can show, so essentially you have the same thing for these models. You have two possible solutions, one where A is expressed and B is 0, and the other one where A is not expressed and B is expressed. And actually what you can show is that for every, so now epsilon scales my diffusion here. And for every epsilon positive, you can find a unique solution to the equations that I showed you. And these solutions are respectively decreasing and increasing, and they are globally attractive. Namely, you start from any initial condition, you will end up on this solution, this unique solution. And what we can show is that when epsilon goes to 0, when the diffusion vanishes, this solution converges towards a discontinuous function, which is actually A expressed up to a specific threshold, and then B, and then 0, and B expressed in the region where A is equal to 0. And so we have a unique boundary here, and we have a very sharp boundary, namely it's discontinuous. And this X star, this position of the boundary, you can characterize it univocally. And it depends on the parameter, but you can compute it only with the parameters. So, so what this means is that the observation that you have a unique solution and that it does not depend on the initial condition is a general property of the kind of dynamical systems that represent homeoprotein interactions in the presence of diffusion. And diffusion, even if it has a small impact, I mean, even if it's very local, it has a tremendous capacity to stabilize the boundary. And actually, even very small diffusions, I mean, the smaller the diffusion, the sharper the boundary will be. So the more stable. But of course, you will have a price to pay, which is that the process takes more time to converge if you have very small diffusion. And what is surprising somehow is that we cannot consider really a diffusing system with very small diffusion. We cannot compare it really to the system with no diffusion because the solutions are very different. One has no real boundary, the other one has a very sharp boundary. And also another conclusion is that a simple mechanism seen in vivo, which is this homeoprotein transfer, could provide a simple and possibly universal mechanism for regularities that we see in development. So I took too much time. Maybe I have 5 minutes? Okay. All right. So no, I would just like to briefly mention, I mean, these works that we did, which is at another time scale, but that kind of consider another aspect of homeoprotein in development, which is the control of timing in development. And this is work that we did with new data collected by Yi, who is a PhD student with Alain and me. And we also worked with Shenju Xu from Taipei and Alessandra Pirani. So essentially, it is relatively well known that homeoprotein, the expression of homeoprotein controls the phenotype of the brain. So this is like, for instance, data by O'Leary and Al and Alessandra also looked at that controls the expression of PAC6. And with a deficient PAC6, you have a change in the boundary between the sensory and between S1 and V1 in mice, with a much enlarged V1 and a smaller S1. And the paper by Shenju Xu and collaborators showed that another homeoprotein, LHX2, controls also the size of the brain. So this experiment was done by knocking out LHX2 at a specific point in time at embryonic day 10.5. And what they observed was that the brain was substantially smaller, and they also observed changes in the number of layers in the brain and also an ectopic piriform cortex. So essentially, it is quite broadly seen that homeoprotein really control the phenotype of the resulting brain. And the question is, how does that occur, and how is timing affected by the expression of homeoprotein? So, so what we know is that the number of cells, so what controls the size of the brain is essentially the number of cells that is produced, the number of neurons. And these neurons are generated through a sequence of divisions and differentiations of specific cells that are called progenitors, and that go through different cell cycles, and they can either make symmetric divisions or asymmetric division to give one neuron and another progenitor, or intermediate progenitors as well. So we have a sequence of divisions and differentiations, and when neurons are generated, they migrate in an inside-out fashion toward the different layers of the cortex. And what is important to control the phenotype is actually a balance between symmetric divisions, which will control essentially the pool of progenitor cells, and the neurogenesis. So the longer this phase, because essentially the duration of the neurogenesis is fixed. So you have a balance between the amount of time you want to duplicate progenitor cells and the amount of time you create neurons. And that's what we're going to study here. So essentially what we did was, so our aim was to write up a model and try to infer timing of neurodevelopment based on observations of the phenotype. And so for that, we developed, again, it's a very simple model as the first one, very naive and based on simple observation, which is that we want to reproduce the divisions and differentiations of cells. So each division and differentiation, occurs at each cell cycle, okay? And it's random, it's stochastic in the sense that the division will occur with a specific probability. So you will have a probability N1 that a radial glial cell, which is a progenitor cell, makes a symmetric division and creates 2. Or another probability that this progenitor gives a progenitor and a neuron. You also have the probability that this progenitor give rise to an intermediate progenitor that eventually makes a symmetric division into 2 neurons. And the last possible division is the loss of neurogenesis capacity of the radial glial cell, of the progenitor, which we say would generate glial cells, but we don't really consider glial cells here. We just consider that the progenitor loses the capacity to eventually create any neuron. So to specify this model, we only need to specify how N1, N2, N3, and N4 depend on time. And so essentially these are the type of curves that we would like to fit. So this is symmetric divisions, then neurogenesis, and gliogenesis. And also we would like to compare, to be able to assess what are the respective number of deeper layer neurons and upper layer neurons. So if we need to fit this model, we would need to find these curves. And Yi did a few experiments to try to access these parameters in the best possible way, considering accessible information from experiments. So first, by looking at the ratio of deeper layer and upper layer neurons at the end of the simulation, we can fix the duration of this phase of neurogenesis for deep layers and upper layer. The switch point from deep layer to upper layer formation, neuron generation, can be also evaluated experimentally, and also she did counts of progenitor cells at different times to kind of compute this curve of symmetric division of radial glial cells. So we need more to fit the model, and for that we use data available from in the literature, in particular The cell cycle duration was taken from the literature. And also there is this remarkable paper by Gao and collaborators in Cell in 2014 that actually reported the evolution in time of the number of progenitors that perform symmetric or asymmetric divisions, and also the proportion of progenitors that doing gliogenesis as a function of the number of clones that they generated. So essentially, we combined all this information and we created a wild-type model. And comparing to this model, mutants will help us understand how these functions are modified based on the phenotype observed. So our first application, and I will try to be brief, is a follow-up of this experiment that I told you about that showed that deleting LHX2 at embryonic day 10.5 yield a very small cortex. So actually, if you delete LHX2 one day after that, you get still a smaller cortex, but by a much smaller amount. You get a smaller and thinner cortex, but you get 6 layers instead of 3. And the deep layer are less affected by this reduction of neurons than upper layers. So the proportions of cells in deep and upper layer is changed. So this was a new experiment from Shenzhou Xu, and we tried to understand what affected that. So for that, there was new data that was collected to kind of fit the number of neurons as a function of time at different times in order to fit our functions. And we proposed different types of scenarios that could account for the changes that are observed in the phenotype. So first, of course, having a smaller volume means that the symmetric division of progenitor, this phase is shorter. And a thinner cortex also means that, we designed four mutants that show different types of shifts of these functions of generation, neurogenesis, and gliogenesis. And we wanted to be able to fit both the reduction in the volume, in the thickness of the cortex, and the changes in the proportions of neuron. And actually, changing these points is not very trivial. Actually, you get changes in both aspects, in all these aspects that are distinct, depending on the way you do it. So we focused on one. There was just one mutant scenario that was valid with the data, which was that actually the switch between neurogenesis and symmetric divisions is advanced, is briefer, and the switch between gliogenesis is advanced by a larger amount than the shift in than the reduction of duration of the symmetric divisions. And so essentially what we got from that was that we had a global shrinkage of the timing of neurogenesis and a disproportionate advance in the gliogenesis phase. And so we proposed that to Shenzhou, and it was tested with marking at different times the glial cells and proliferating cells. And indeed, when comparing to the wild type, it was observed that at a much earlier stage, we had gliogenesis, compared to the wild type mice, which was which kind of validated this prediction. And I guess I no more have time to speak about compensating cell death. I will just end up by summarizing that I presented a few thoughts and models, mathematical models for neurodevelopment, and I discussed the fact that the diffusion of the non-autonomous activity of Homeoprotein could provide a solution to create smooth boundaries with reliable locations, and that this is a general mathematical property of competitive systems with graded Q, monotonic Qs. And I also presented, but very briefly, a stochastic model of deviation and differentiation that allows more or less inferring information about the timing of morphogenesis and its impact on brain phenotype. And I would like to thank the collaborators. This is Cristobal, who worked on the Arealization project. This is Yi, that are the main person, and Alain, Shenjushu, and Alessandra, with whom we work.